Pues muchísimas gracias, doctor Moya. Es un placer estar aquí. Siempre venir a España para mí es como venir a casa y realmente es un orgullo para mí poder empezar este curso. Así es de que voy a cambiar al inglés, porque si no, Dobry no me entiende y yo quiero que me entienda porque vamos a tener una muy buena discusión aquí. Lo que yo les pido a los ponentes es que hablen didácticamente. So I'm going to ask each and every one of the speakers to be didactic, to be clear, and to be on time, so that we can have enough discussion at the end. And we will sit down at the end and answer any questions, get any tomatoes that we may receive from the audience. And I ask the audience to please be proactive. If you feel that you do not want to ask a question because you fear the English is not going to be good, don't worry about it. I'll translate it for you. And what I need is everyone to be active. We're a small group, but usually small groups are the best groups. So don't be shy, ask your questions, and enjoy the presentation. What we are going to be doing here is going to exactly to find our way in translation, not only in translation from English to Spanish and back to English, but also in the mechanisms of atrial fibrillation. And we're going to go from the gene to the bedside. And that's why I put together a group of investigators from Europe as well as the United States who are going to be dealing with all of these subjects. Now, your training, in the majority of people in this room perhaps, or in this, sorry, that's Paloma, sorry. Sorry about that. The majority of people in this audience and in the Congress actually deal with these things, and we don't have to tell you about this. We're not going to tell you about this. We're going to tell you about basic mechanisms of cardiac arrhythmias, and we're going to try to translate into what you see in the patient when you do a mapping with the electroanatomical system, the cardiac system, or whatever system you use, and also when you use electrograms. It's very important to realize that using one single electrogram, you will never find a rotor, you will never find a mechanism. You need high resolution, and we should start from that. And we're going to be dealing at the gene, at the cellular, at the molecular level, and at the organ level with the vicious circle of atrial fibrillation. We're going to start with the triggers, and then we're going to look at the substrate, and we're going to be looking at rotors. And then we're going to look at how these rotors generate high-frequency excitation of the atria, and that maintain the atrial fibrillation. And then once we do this, we're going to be looking at how sustained atrial fibrillation leads to remodeling of the atria. And when this remodeling of the atria occurs, it modifies the triggers, and it also modifies the substrate. The title of this presentation is to talk about rotors, but we're going to be dealing with all things related to rotors, particularly what triggers those rotors, what maintains it, what is the substrate in the atrium that keeps these rotors lasting for many, many years in some patients. And we're going to be seeing how these rotors are recorded and how they are analyzed. So sit back, relax, enjoy the show. And I'm going to start by introducing the first speaker, but let me tell you about the speakers. Dr. Dobrev is going to be talking about intracellular calcium and the mechanisms of fibrillation in terms of triggered activity, delayed after depolarizations, early after depolarizations. Then we're going to move on to the substrate, and Dr. Ricardo Caballero from the Complutense is going to be talking to us about ionic mechanisms of remodeling. They have done beautiful work on ionic mechanisms of remodeling, and you're going to enjoy this. Then Sandy Pandit, who is at the University of Michigan, is going to give you the basics for rotors. What is a rotor? How is a rotor initiated? What maintains it? What are the properties of the substrate that are needed to maintain those rotors? But he's going to do that at the very basic level. Sandeep is a biomedical engineer who has strong training in biophysics and who knows how to think about rotors in terms of modeling. Then we're going to have a break, and hopefully we're going to make that break short so we can take more time for the presentations. I will be talking about how rotors interact with the substrate and what is it that the remodeling does to the substrate. And then Omer Berenfeld is going to be telling us about how to map these animals that are the rotors. And then Juan Tamargo, who is very well known to all of you and to all of us, is going to tell us about the pharmacology of atrial fibrillation, and hopefully he's going to tell us about the pharmacology of rotors if he is interested. And then finally, Felipe Atienza is going to tell you the latest about the radar trial, which was very successful, and it's a very important trial here in Spain that was looking at how dominant frequency can be used to terminate atrial fibrillation. So without further ado, I want to thank the organizers. Particularly, I want to thank Nicasio for the invitation. I'm really honored that you chose me for this, and I'm delighted, and I hope that you're not disappointed by the presentations. And so my first speaker is Dr. Dobromir Dobrev, who's going to be talking to us about intracellular calcium and the mechanisms of atrial fibrillation. Dobry.
Thank you very much, Pepe. Good afternoon to everyone. I also would like to thank the organizer for inviting me to present here, and in particular to Pepe, who I know now for many, many years. The title of my talk is dealing rather with the mechanism at the cellular level, and I will present you data, hopefully to convince you that you can learn about the clinical mechanisms of atrial fibrillation if you study single cells. Just the conceptual framework, Pepe already mentioned it, what we are talking about. I found a little bit the color strange, but never mind. We are talking about as a fundamental mechanism, re-entry as a maintaining mechanism and ectopic activity. Re-entry requires a substrate, frequently trigger at least an initiator to initiate re-entry. And now we know also that there is a very important role for remodeling processes in the atria, which change the function and the structure. We know a lot about the ion channel changes, and also the structural changes, particularly fibrosis, and I'm sure that we will hear a lot about this in the subsequent talks. What we know very well now is the substrate at the molecular level, the substrate due to the cardiovascular diseases, and also the substrate produced by the high atrial rate itself. What is less well known is about the triggers. What is the mechanism here for re-entry? Just a scheme. I don't want to go to the details, but ion channel remodeling is supposed to play an important role, at least at the beginning, where the frequency is increased. Let's say, see here the 300 to 400 beats per minute, and ion channel remodeling resulted change in the shape of the atrial action potential that's shown here. You see that the early repolarization is prolonged, the later repolarization is abbreviated. There's a lot of ion channel changes that I don't want to touch here, and the result is that the wavelength is reduced, more re-entry circuit can be accommodated, and this is a vicious circle. There's very important role for fibrosis in later stages, probably in patients with atrial fibrillation or such who developed atrial fibrillation in the structurally remodeled heart. We know now that it is very important to look at the fibroblasts, which were neglected for many, many years in our field, and now we know that the differentiation to myofibroblasts is extremely important, and ion channels appears here to play also an important role. I was particularly asked today to discuss with you the potential role of the so-called delayed after depolarization, which are supposed to contribute to ectopic activity, which could do two things. First, could be the trigger to initiate re-entry in a arrhythmogenic substrate. And if the frequency is very high and repetitive in nature, it could be even the driver for atrial fibrillation. And such a mechanisms, DAD-based mechanisms, is supposed to be one potential mechanisms for the focal sources observed in the area of the pulmonary veins. What is the concept? Now I go to the cellular level. What you see here is schematic, a cardiomyocyte. This is the membrane. This is the intracellular calcium store, the so-called sarcoplasmic reticulum. Delayed after depolarization can be studied at the cellular level. This is the difference to re-entry, which is a macroscopic mechanism. The central role here is for the so-called ryanodine receptor channel, which is the channel releasing calcium in response to stimulation during systole and provide the calcium which is required for contraction. This calcium during the diastole is removed by the so-called sodium-calcium exchanger. This is a protein in the plasma membrane. This is pumped out for each calcium outside 3 sodium ions into the cell. And if this is frequently enough and much calcium is released during the diastole, this produces delayed after depolarization. You see here oscillation in the resting membrane potential. And if the threshold is reached to activate the sodium channels, you can have triggered action potentials, and this is the so-called triggered activity which could promote atrial fibrillation. Are there evidence that this happens in myocytes from patients with atrial fibrillation? This is indeed the case, and this is now for many, many years. The first demonstration was in 2004. What you see here, I don't want to go into the details, but these dots show here represents the release of calcium to ryanodine receptor clusters. And what you see here is a myocyte from a patient in sinus rhythm. Here is a myocyte from a patient in atrial fibrillation. And you see that the numbers of dots is much more frequent in atrial fibrillation as a control. Here are the statistics. These are the so-called calcium sparks, and also the so-called calcium waves appears to be increased. What could be the basis for this mechanism? Is this a dysfunction of the ryanodine receptor channel itself? What is well established that these ryanodine receptor channels are regulated through phosphorylation. And this phosphorylation is shown here with the P symbol can be at 2 different sites which are now well established by 2 different kinases, the so-called calcium remodeling kinase and the pKa, and hyperphosphorylation of the channel increases the sensitivity to diastolic calcium in the channel open during the diastole, producing the so-called calcium leak. If you take tissue from patients with chronic persistent atrial fibrillation as shown here, and you measure with the so-called Western blot the protein levels and the phosphorylation levels at the both sides, you will find that these channels are hyperphosphorylated at 2 phosphorylation sites, and this is expected to increase the open probability and to produce the leak. If you take the channel and measure the single channel open probability as shown here, you see this is the closed state. If the channel open, this is shown by this peak here. This is sinus rhythm, chronic atrial fibrillation channels from patients put in the artificial system in order to be studied. I don't want to go to the details, but you see that on the right side, the frequency is much, much higher. And if you study it as a function of calcium ions in cytosol, here the open probability, you see that the open probability is increased in patients with long-standing persistent atrial fibrillation, which suggests that this channel is dysfunctional, is not closed during diastole, which is physiological, and produces calcium leak, which can cause delayed after depolarization. This is a complex protocol where you can study the so-called calcium leak in intact myocyte. I don't want to go again into the details, but what you see here, that you measure voltage and calcium movements in a single human atrial myocytes in the same time, and if you claim the myocyte as minus 80 millivolt to simulate the diastole in the absence of sodium and calcium ions, so there is no transmembrane fluxes in the myocyte. And use a drug, what you know as an anesthetic, tetracaine. Tetracaine blocks ryanodine receptor channels, and the decrease in diastolic calcium, as shown here, is proportional to the calcium leak in these myocytes. And at the end of the protocol, we use caffeine to deplete the sarcoplasmic reticulum and to be able to relate the leak to the load of the myocyte. And if you calculate this, the SR calcium leak of the myocyte is much, much higher in atrial fibrillation. If you look now whether the phosphorylation of the channel plays a role, here we use the inhibitor of the calcium calmodulin kinase. We can prevent the leak, the difference between sinus rhythm and long-standing atrial fibrillation, whereas if we block the protein kinase A, we were unable to remove this. And even if we normalize to the load, the leak to the load of the individual myocyte, the differences between control and atrial fibrillation persist, and only the COMK2 inhibitor can remove the difference. Why is this? We believe that the mechanism is complex, but if you take tissue from patients, atrial tissue, and look for the expression and the so-called autophosphorylation of calcium calmodulin kinase, which suggests is an index of activity, you will find that the expression is much higher and the activity of COMK2 is higher, probably hyperphosphorylate ryanodine receptor channels, making these channels leaky and leading to delayed after depolarization. The question is whether these calcium releases, as shown here, produces, in fact, depolarizing inward currents through the sodium-calcium exchanger. This is shown in this figure. Here in sinus rhythm, this is complicated protocol. You study here calcium transients, the calcium movements in the cell, and the resulting sodium-calcium exchanger current. That if you stop the stimulation, claim the myocyte at minus 80 millivolt, you can see whether the ryanodine receptor channels remain closed. This is not the case. Sometimes they open even in sinus rhythm, and they produce a sodium-calcium exchanger current. If you look at long-standing persistent atrial fibrillation, the frequency is higher, as shown here. The latency, that means that the leakiness is start much earlier after the regular stimulation, as shown here. And also the amplitude of these events is much higher, and the resulting currents as shown here is in atrial fibrillation much, much higher. This is a direct demonstration that this SR calcium leak produces depolarizing currents which can produce delayed after depolarization. And this is studied in the subsequent slide. Again, here you see the regular calcium transients stop the stimulation. Here are the spontaneous calcium releases through dysfunctional ryanodine receptor channels. And what you see here are delayed after depolarization. Now here is voltage measurements. These are the action potentials. And if you repeat this experiment in myocytes from patients with chronic atrial fibrillation, you see that here you have much frequently releases of calcium, and these cause oscillation of the voltage, which is analyzed here. The membrane voltage difference in response to calcium is much, much larger in chronic atrial fibrillation as in sinus rhythm. And this is the first demonstration of delayed after depolarization, to my knowledge, in the human heart at all, not just in the atria. This is a slide I want to skip. What I showed you is that delayed after depolarizations occur in myocytes from patients with long-standing persistent atrial fibrillation. The issue here is that we have hyperphosphorylated ryanodine receptor channels, increased diastolic calcium leak, activates the sodium-calcium exchanger. There is a sodium load because of these mechanisms, delayed after depolarization that I showed you, and ectopic activity. The question is, how can ectopic activity exist if the frequency is so high in the patients on long-standing persistent atrial fibrillation? And I don't want to discuss this issue because this leak is not doing just DADs, but also calcium-dependent remodeling, contributing even to the structural remodeling of the atria. But the issue here is that we don't really know whether this ectopic activity I showed you at the cellular level is the cause of atrial fibrillation, or is rather the consequence of the high atrial rate itself. And what we did there, and it was for many, many years, very difficult study, we repeated this study in myocytes from patients with paroxysmal atrial fibrillation. They had the last episode of atrial fibrillation is a median on 15 days. They don't have the consequences of the arrhythmia itself. And what we did here, you see, we took again right atrial appendages of these patients. All the data I showed you are right atrial appendages. It had nothing to do with the left atrium or with the pulmonary veins, because we don't have access to this tissue. And what you see here, here's the masses, here's the patch pipette, and here's the myocyte. What we first did is we looked for the so-called electrical remodeling, defined as an abbreviation of the action potential, as shown here at the cellular level. We didn't see any changes in the APD 20, 50, or 90, no changes in resting membrane potential, and also no changes in action potential amplitude. So the classical indices of electrical remodeling, which are typical for chronic or for persistent atrial fibrillation, are completely absent here. And we can ask the question whether we can see the same spontaneous releases of calcium producing DADs and sodium exchanger current, and it is shown here. The same protocol here, you see calcium, here you see current measurement. This is the sodium calcium exchanger current. This is much more frequent and much larger frequency in myocytes from patients with paroxysmal atrial fibrillation. They don't have AF-related remodeling, at least based on the electrical properties. We can look for DADs, now again, calcium measurements, and here are action potential where you can measure the voltage change, and you see that in patients with paroxysmal atrial fibrillation, the amplitude of these spontaneous calcium releases of the leak are much higher, and this produces, of course, a bigger change in the voltage in patients with paroxysmal atrial fibrillation as compared to controls. What is the mechanism? We were interested to see if that is the case, whether we see the same molecular changes also here. What we found is that, of course, the SR calcium leak measured with this protocol I showed you a few slides ago, and we found that the leak is higher. What is interesting and different to patients with long-standing persistent atrial fibrillation is that the SL calcium load, the amount of calcium within sarcoplasmic reticulum, was significantly increased, almost doubled. And just to remind you that this SL calcium load itself is sufficient to produce spontaneous calcium release in DADs, and this is the mechanism how digitalis glycosides produces ventricular arrhythmias due to SL calcium load. If you have high dose of digitalis, you can induce arrhythmias, and this is the mechanism, is increase in sarcoplasmic reticulum calcium load. We looked then for the expression and phosphorylation of ryanodine receptor channels. I personally was surprised to see that the channel is not hyperphosphorylated. That was our hypothesis. What we found is rather that the expression of ryanodine receptor channels was threefold higher in patients with paroxysmal atrial fibrillation. We have no idea what is the mechanism, but this is the result. And if you look at the single channel activity, whether the channels open much more frequently at diastolic calcium, this is shown here, this is indeed the case. We have more ryanodine receptor channels, they have higher open probability, and this is associated with an increase in sarcoplasmic reticulum calcium load. And if you here are data whether we looked what about the function of the calcium ITPase, which take up calcium into sarcoplasmic reticulum. We asked the question whether the activity is increased, but we found that the expression was decreased, whereas the regulator phospholamban had a higher activity. This decrease could be offset by this increase. This data was not conclusive based on biochemistry. And here we used a very complicated protocol, which I don't want to go into the detail, but just focus on this data here. This is an index of the SERCA activity. What we found is that the activity of SERCA is in these myocytes significantly increased as compared to control. And want to summarize as a conceptual framework here, what I presented you is that hyperphosphorylation of ryanodine receptor channel or increased expression and increased SR calcium load can cause increased frequency of diastolic SR calcium releases, frequently called SR calcium leak. This is non-physiological. Normally, these channels are almost all closed during diastole. This produces inwardly directed depolarizing sodium calcium exchanger mediated current. This changes the voltage. The response of the voltage to calcium is increased, at least in persistent atrial fibrillation. This can go as DADs, a trigger action potential producing ectopic activity, and this scenario is very likely to contribute to the atrial arrhythmogenesis by patients with paroxysmal atrial fibrillation. And I didn't show you the data, but we have preliminary data suggesting that this diastolic calcium leak can produce persistent leak during diastole, produces calcium-dependent remodeling, and changes conduction velocity, and also can cause repolarization changes, increasing the dispersion of refractoriness. And there are now emerging data to suggest that SR calcium leak may contribute also to the determinants of reentry and have a much larger impact as we suspected initially. In conclusion, sarcoplasmic reticulum calcium leak together with cellular DADs and triggered activity appears to play an important role in patients with paroxysmal and long-standing persistent atrial fibrillation. In patients with paroxysmal AF, these releases in the DADs may be part of the cellular mechanisms of the clinically observed focal ectopic activity that triggers and may even maintain AF recurrences in many patients. And in patients with long-standing persistent AF, the continuous diastolic SR calcium leak causes calcium-dependent remodeling and contributes to the stabilization, likely contributes to the stabilization of reentry and AF maintenance. I thank you for your attention.
Very nice, Dobry. Thank you very much. I think we have time for questions. Stay on the phone. Yes, there's a question there.
Yes,
Can you identify yourself?
myocytes. But you also show that you have an increase in the SR calcium content. So why you have an increase in the SR calcium content if you have a leakiest pathway out of the SR?
This is a difficult question, a question what we always get. You have to consider that we studied this myocyte artificially, and we do a lot of different stuff. The issue is that you would expect that if you have leak, that the load should be decreased. In patients with long-standing persistent atrial fibrillation, you have leak and the load is normal. Here we even have an increase. Why these changes are and why this steady state is so different as we know from physiology, I have no idea. But the issue is that we used a computer model to look whether the leak itself or the load are sufficient to produce these DADs and spontaneous calcium releases, and it appears that each of these factors have an independent contribution to the DADs. In the combination of both, as we find in a paroxysmal atrial fibrillation, is very likely pro-arrhythmogenic. But we are working now on the mechanisms to see why is the open probability of ryanodine receptor channels increase in paroxysmal AF and why is a calcium load. But I cannot answer the question.
So, Dobry, I have a question. As you know, there has been a recent paper by Downers, written on ventricles, ventricular fibrillation, and they demonstrate. Can you hear me?
You can come here, baby.
Okay. So there has been a recent paper by Don Bears and Ripplinger that just appeared in Circulation Research, in which they studied simultaneously with calcium and voltage right on the ventricle what is happening to DADs, actually mostly what's happening to alternans. And the most important question that for me they were asking was what's happening during ventricular fibrillation. And they showed that during ventricular fibrillation, there's absolutely no release from the ryanodine receptor because it's refractory. So this brings me to the question, Is the DAD that is produced by the CAM kinase activation of the RYR2 phosphorylation of the RYR2 really in atrial fibrillation having a significant role when the frequency of atrial fibrillation is 5 to 8 hertz? Can you justify the release of calcium at that level?
No, clearly no, and this is a very important issue. Whereas in paroxysmal atrial fibrillation, we have studied the myocytes in vitro at the same frequency they see in vivo, at least if they don't have fibrillation episodes. This is different for chronic atrial fibrillation. We took out the tissue from 8 hertz and studied in vitro at 1 hertz. Of course, we see now that there is a dysregulation, and we can study, and I produced to you, I hope, a very nice story, but I can imagine exactly what you are saying, that at 8 hertz, the channel is completely refractory, and there is no role for this mechanism, at least in patients where they never have sinus rhythm. You know, persistent atrial fibrillation, you know, is defined as longer than 7 days. Right. So long-standing persistent, where we are sure that is probably not the mechanisms, but in those where they have for several weeks AF, they have some episodes, could be that there is during sinus rhythm, this helps to reinitiate the arrhythmia. There's probably a role for that.
There's still room for lots of questions, but let's keep it to the end, because then we will have a nice discussion after my presentation, I hope. So let's thank Dr. Dober for his presentation. So the next presentation is by Ricardo Caballero. Ricardo, please come forward. He's going to be talking to us about the molecular mechanisms of ionic remodeling. And he's going to be talking about different ion channels. They have been studying the entire spectrum of ion channels in the laboratory of Ricardo together with Eva Del Pon and Juan Tamargo, and they have published some absolutely magnificent studies. Some of them published in Circulation, PNAS, etc. And I'm really proud that Ricardo has accepted my invitation. Ricardo, please.
Thank you very much, Professor Jalife, for these words. And first of all, I would like to thank the organizers, Dr. Nicasio Perez Castellano, and of course, Professor Jalife, for giving us the opportunity to be here in this excellent meeting. And I would like to recognize my honor for being here personally and transmit the best regards from Professor Del Pon that finally couldn't be here. I would like to start with this slide telling that it's considered that atrial fibrillation is a progressive disease that in many patients starts with recurrent self-terminating episodes of very short duration, by definition less than 7 days, that in most of the patients progress to more persistent and permanent forms with recurrent episodes of longer duration that may require a medical intervention to terminate, or even when these episodes never terminate spontaneously or with any medical intervention. And that is the permanent or chronic form of atrial fibrillation. This progressive pattern suggests that there is any mechanism that is promoting the maintenance and recurrence of the arrhythmia. In the last years, we have obtained many pieces of information, and now we are firmly convinced that atrial fibrillation is producing important changes in both the structural and electrical properties of the atria that lead to the arrhythmia maintenance and recurrence. That is a process well known that is called structural and electrical remodeling. I'm going to focus in the electrical remodeling, and I would like to start with this famous figure that is the first evidence that atrial fibrillation was producing changes that leading to the maintenance of the atrial fibrillation episodes. This is a very famous figure published by the Professor Alesi group in Circulation in 1995, and they measured the atrial fibrillation inducibility in a goat model of rapid atrial tachypacing. And what they saw is that when they apply a rapid burst pacing, in those animals that were all the time in sinus rhythm, the episodes was very short, only lasting some seconds. But when the burst pacing was longer, you see the episodes last longer. And even when they applied the bursting after 2 weeks, for 2 weeks, the episodes were perpetual. The AF became sustained and no longer terminate spontaneously. So they entitled this work, Atrial Fibrillation Begets Atrial Fibrillation. They, in this paper, also measured the refractory period, and they found that as the atrial fibrillation episodes were longer, the refractory period was shorter, and they demonstrating that atrial fibrillation was shortening this parameter. The same effect, the same results have been observed in humans, and many clinical studies have demonstrated that atrial fibrillation patients have a shorter refractory period than sinus rhythm patients. But what happened at the cellular level? In this part of the slide, I'm showing two action potential recordings made by the group of Professor Dobrev that comparing the action potential in myocytes from sinus rhythm or chronic atrial fibrillation patient. As is evident, the duration of this action potential is quite different, being much shorter in the CAF patients, with a perfect correlation with the shortening of the refractory period. So atrial fibrillation is producing a shortening in the action potential. To understand how action potential can be shortened, we should remember how these action potentials are generated. As you know, the action potential is a reversible change in the membrane potential as a consequence of changes in cell permeability to different ions, mainly sodium, calcium, and potassium. The polarization phase is due to the entry of the sodium through voltage-gated sodium channels that you have here in the table. But as you can see, the repolarization phase is much complex. It's a fine equilibrium between the entrance of calcium through voltage-gated calcium channels and the exit of potassium through several potassium channels. We have voltage-gated potassium channels, mainly 4, the one that generate the transient outward current, ITO, and the delay rectifier channels classified depending on the activation kinetics. The ultra-rapid, that is atrial specific, this current is called IQR. The rapid, the IQR, generated by the famous air channels, and the slow delay rectifier IKS. At the end of the repolarization, we have activated 2 inward rectifier channels, the IK1 and the current activated by acetylcholine, that is also atrial specific. These currents are generated by KIR2 and KIR3 channels, respectively. So, taking all this data in mind, we should think that a shortening of the action potential duration can be obtained by 2 very easy mechanisms. Either decreasing the entry of positive charges, like calcium, or increasing the exit of potassium from the cell. So, in the next slides, I'm going to show some of the data available about changes in these ion currents that are produced by atrial fibrillation. I'm focused in the data available only in human atrial preparations, and all of them are being obtained in human atrial myocytes enzymatically isolated from right atrial appendages from patients in sinus rhythm and chronic atrial fibrillation undergoing cardiac surgery. The first paper we found, to my knowledge, is this one. It was published by David Van Bangoner and Jean Nerbonne in Silk Research in 1997. And surprisingly, they found that two outward potassium currents, like the ITO and the IQR, were decreased, not increased like they were expecting. You see, the ITO is corresponding to this rapid activating and rapidly inactivating component, whereas the IQR is corresponding with this component at the end. This decrease was due to a reduction in the expression of the channels in the membrane, as can be observed in these yields. And the results were specific, since other potassium channels like Kv2.1 were not altered in these preparations. So the thing is that we cannot explain the shortening of the action potential by these results, because a decrease in the potassium currents would produce a lengthening of the action potential duration. So they interpret these results as a compensatory mechanism trying to prevent an excessive shortening of the action potential. 2 years later, the same group published this important paper showing for the first time that chronic atrial fibrillation is decreasing the calcium current. Now we have the first evidence of a current that whose change is producing a shortening of the action potential duration. After this paper was published, many other groups, some of the references are included here, have observed the same results, this reduction of the calcium current in chronic atrial fibrillation patients, and now it is considered a hallmark of atrial fibrillation electrical remodeling. A couple of years later, from the paper from Wagoner and Jean Nerbonne, the group from Dresden, here represented by Professor Dobrev, demonstrated that the inward rectifier current activated by acetylcholine, which is here represented by the current increased by an acetylcholine analog, which is carbachol, which is represented by white circles, is very large in sinus rhythm myocytes. However, in atrial fibrillation myocytes, you see the amplitude of this component is very small. Conversely, some years later, they described that in the atrium there is a constitutive component of this current, of this AKCH, that is very small in sinus rhythm myocytes, as can be observed when they perfused with the channel blocker that is tertiapine. You see the very small inhibition reached with tertiapine, but you see now in chronic atrial fibrillation the decrease of the current is very high, very large. So they conclude that this constitutive component of the IKCH is much higher in chronic atrial fibrillation myocytes. I'll remind you that this current is expressed only in the atrium, so this is considered now a promising drug target. Let me come back to the paper from the group from Dresden in 2001. They also described that the IK1 current, that is here represented by the black circles, is quite small in sinus rhythm myocytes, but you see is very high, very large in atrial fibrillation myocytes. We think that this change in the IK1, this increase in IK1, is a very important finding because of the special features of this current. You see, in this part of the slide, we show the IK1 recorded by using an action potential shape protocol. You see, this is the amplitude of this current that is close to zero in the action potential during the action potential, but only at the end of the action potential, the amplitude of this current is markedly increased. So we can say that this current is participating in the final phase of the repolarization, not only in the control of the membrane potential, at the resting membrane potential. So we can think that a huge increase of this current at the end of the repolarization would be contributing to the shortening of the action potential. Moreover, several authors have demonstrated that in several species, IK1 density is larger in the left than in the right atrium. And this could be producing the differences in the activation dynamics, the activation frequency that, as Professor Khalife will mention later, is faster in the left than in the right atrium in atrial fibrillation. So we think that IQ1 increase is a very important change produced by atrial fibrillation by its own. So we demonstrated that this increase in IK1 was also produced in left and in right atrium, confirming the results published by Boyd and co-workers in 2010. And we also see that this increase was in the outward and in the inward component of the current IK1. And we saw that the increase was bigger in left atrial appendages, suggesting that this increase in atrial fibrillation myocyte was contributing to this, to the heterogeneity between both atrias. So, so far I've shown that we have changes in calcium and potassium channels induced by atrial fibrillation that correlates perfectly with the shortening of the action potential and refractoriness. So things are very easy. But with time, we have learned that things are not so easy. Let me show this couple of slides telling how things can be complicated. In normal atrial tissue, some authors, it's a paper published by Dr. Natel group in 1993, they show that at least 3 types of action potential can be recorded in atrial tissue. One action potential with this very prominent spike of fast repolarization followed by this huge plateau. Another action potential without this spike and without this prominent plateau, and those with an intermediate pattern. Another piece of complication has been demonstrated both in experimental models and in humans, demonstrating that, as I mentioned before, that atrial fibrillation reentrant sources are faster in the left than in the right atrium in several types of atrial fibrillation, postoperative, paroxysmal, and persistent atrial fibrillation. So if things are complicated, we can think that how this heterogeneity, both in physiological and pathological conditions, can modify the electrical remodeling that was observed some years before. So we made 2 questions. The first one, how would electrical remodeling interfere with the physiological electrical heterogeneity within the atrium? And the second question was, would the electrical remodeling affect differentially to the ion currents in the left and the right atrium? To answer these questions, our group was implicated in an ambitious project with well-known researchers in this field, in a project coordinated by Professor Almendral that was funded by CENIC some years ago. This project was very important for us because it allows us to get human samples from right and left atria to isolate myocytes and to get very important results. The samples were obtained from the cardiac surgery and cardiology services of the Hospital Gregorio Marañón. I'm going to show you some results we obtained in this project. The first thing we saw is that in SINUS RHYTHM, in cells from sinus rhythm patients, we observed at least 3 types of cells, depending on the outward potassium current that is predominant in each cell. We have these currents with an ITO, transient outward current predominant, more or less 30% of the cells. Other cells with the IQR predominant pattern, only IQR, not ITO, with 20% of the cells, and an intermediate patterns with both currents, that is the rest of the cells. It was the same pattern in left and right atrium in sinus rhythm, but importantly, these percentages were modified differentially in the left and in the right atrium. You see in the left atrium was a decrease in the ITO predominant cells. However, in the right atrium, a decrease in the ITO, but an increase in the sustained and the IQR current was produced. We also observed that the reduction of the transient outward current and the IQR that was published by Van Wagoner some years ago was not homogeneous either in right and left atrium. You see in the ITO, we can observe that the decrease is much bigger in the left than in the right atrium. So we have a gradient of ITO density that was not present in sinus rhythm cells. However, in the IQR, we saw that in sinus rhythm, the density was bigger in right atrial cells. However, in cuff cells, we abolished this gradient. So we observed that chronic atrial fibrillation was producing this gradient, abolishing this gradient, and we now have more heterogeneity than was before under sinus rhythm conditions. But the most important result we obtained in this project was this. We observed that in chronic atrial fibrillation myocytes, a huge increase in the slow delay rectifier current, the IKS, was produced in both atria, left and right, as a consequence of an increase in the subunits of forming the channels. We think that it is a very important result also because, especially for the features of the IKS. First of all, due to the very slow activation and inactivation kinetics, IKS accumulates and its amplitude is bigger at high than at very low frequencies. So, and this difference is especially observed in the chronic atrial fibrillation situation. We have to consider that, as Professor Dobrev told us before, during atrial fibrillation, we can obtain a frequency of stimulation of 8 or 10 Hertz. So this, under this condition, this current would contribute to the control of action potential duration, more importantly, at a very low frequency. So we think that could be desirable to obtain a drug that would block IKS in a frequency-dependent manner. But we have this drug. Our group, several years ago, described that propafenone produced a frequency-dependent block of IKS. So propafenone could serve as a model for designing drugs that produce a frequency-dependent block of this current. The second feature of the IKS is high sensitivity to the stimulation from beta receptors, from beta-adrenergic receptors. This project, we also saw that the expression of beta-1 receptors is greater in chronic atrial fibrillation atrial samples, especially in the left atrium. We consider that IKS is increased by the stimulation of beta-1 receptors. We have, in these patients, we have IKS increased by 2 different mechanisms. One, by the cuff itself, the chronic atrial fibrillation itself, and when the beta-1 receptors are stimulated. So we obtain the following results. We see when in the coronary atrial fibrillation myocytes, we stimulate the beta-1 receptors with isoproterenol, we see a huge increase in the density of the iCaL that is greater in the left than in the right atrium, an effect that is completely blocked by atenolol, which is a beta-1 receptor antagonist. So we think that this is a very important result. So, but with all this data, what are the consequences at the action potential level? So if we stimulate beta-1 receptor in myocytes from sinus rhythm patient, what we see is that isoproterenol prolongs action potential duration as a consequence of its increasing effects on the calcium current, so nothing new. However, when we apply isoproterenol in chronic atrial fibrillation myocytes, we see that isoproterenol is decreasing the duration of the action potential. We have to consider that in these myocytes, calcium current is very small and iCaL is very big. So now we can see that isoproterenol is shortening the action potential, probably due to its effects on the iCaL. So let me finish my presentation with a couple of slides commenting on the molecular mechanism underlying these ion channels changes I've shown so far. This, as Professor Dobrev described before, these changes are mediated by an intracellular calcium overload due to the high atrial rate. This mechanism has been studied extensively for the calcium current, and we are going to show some data about that. The mechanisms of the calcium current downregulation are very complex, and this slide tries to summarize some of these mechanisms. Some of them are at a transcriptional level, decreasing the expression of the calcium channel in the membrane. Another mechanism are post-transcriptional, and some authors have described that there are a role for an equilibrium between dephosphorylation and phosphorylation of the channel. Some other authors have proposed that there is an increase in the protease activity, and very recently some authors have proposed that microRNAs can play a role in this downregulation of the calcium currents. These microRNAs are small non-coding RNAs that regulate gene expression by binding to 3 UTR region of the target mRNA, promoting its degradation or blocking translation. There are several microRNAs that have been demonstrated to change in the atrial fibrillation setting. Indeed, some of these microRNAs are involved in the electrical remodeling. Other microRNAs have been demonstrated to play a role in the structural remodeling. Our group was focused on MIR21, that is one of the microRNAs that is most upregulated in atrial fibrillation patients. So far, MIR21, since it's highly expressed in cardiac fibroblasts, it has been proposed that it's playing a major role in fibrosis and structural remodeling. However, its expression in cardiac myocytes has been a matter of debate. So we surmise that MIR21 is probably expressed in human atrial myocytes, playing a role in electrical remodeling. The first thing we did was to isolate myocytes and study the expression of this microRNA in these myocytes without fibroblasts in the medium. So we observed that MIR21 is expressed in human atrial myocytes, and this is more expressed fourfold in chronic atrial fibrillation myocytes. Moreover, the expression of this MIR21 is perfectly correlated with the expression of the subunits of the calcium current and with the L-type calcium current density. And most importantly, we saw that the expression of MIR21 in cultured atrial cells decreases the current density, calcium current density, and produce voltage-dependent effects identical to those produced by chronic atrial fibrillation. So we proposed that apart from the effects of MIR21 on fibrosis, it could play a role in the electrical remodeling through an effect on the calcium current. So it can be possible that atrial fibrillation is increasing MIR21, and this is a link between structural changes and electrical changes in the same way that this has been described for TGV beta, etc. So let me conclude with these conclusions of the talk. That is electrical remodeling, which is mainly characterized by a shortening of action potential duration and refractoriness, is characterized by a decrease in the calcium current, an increase in IK1 and constitutive IKCH. They are very important findings. And we propose that an increase in IKS is also important due to the special features of this current. Finally, molecular mechanisms underlying ionic remodeling are very complex, as I mentioned, and microRNAs could be involved in this mechanism. Let me conclude with the most important slide, that is the one I used to thank all the people that made possible all these things, especially to the directors of the group, my mentors, Professor Tamargo and Professor Del Pont, the people in the hospital that gave us the samples, and the institutions that give us the money, of course. Thank you very much for your attention.
Very nice, Ricardo. Wonderful idea with the MIR21 affecting both fibrosis and ion channel remodeling. Any questions for one or two short questions? I have a question, Ricardo. In the effects of isoproterenol on IKS, I noticed in your superimposed action potentials, not only that in the calf there was a shortening of the action potential, but there was a slight hyperpolarization. What is the effect of IK1 there?
Yeah, the isoproterenol is blocking the IK1 in this paper we published, but we were surprised by the fact that the inhibition of the current is only at negative potentials. We have no clear explanation for that effect, and at this concentration, we used a very small concentration to give very specific effects on the currents, and we don't know how the effect is only on the negative potential. So we don't expect a huge effect on the resting membrane potential. What we see in this action potential is calf myocytes are hyperpolarized compared to the sinus RHYTHM myocytes.
So what is the mechanism?
An increase in IK1.
Okay.
We saw that
the effect actually may be a synergistic effect between the increase in IK1 and the shortening of the action potential.
Yeah, for sure.
That's good. Very nice. So at the MIR level, tell me, what is MIR doing to fibrosis?
MIR21.
How is it affecting fibrosis?
MIR21 has important targets in fibrosis. The most important one is SPROTI. SPROTI one that is making everything for fibrosis. And it's very, very well known, the pathway that is moving the fibrosis to upwards. And this is the first piece of evidence of MIR21 related to atrial fibrillation was its role in fibrotic pathways.
I'm going to leave my questions for later, but anybody has a burning question that would like to ask? Yes, please.
So is there any room for the forward mode sodium calcium exchanger current in your action potential repolarization and the differences you see between the sinus rhythm and the atrial fibrillation? Any room for the forward sodium calcium exchange current?
Yeah, we made experiments trying to isolate the sodium calcium exchanger, and we didn't see changes, importantly by isoproterenol, in our conditions that are not optimized to record this kind of channels. But of course, it can be participating in the effects of the isoproterenol, of course.
Thank you, Ricardo.
Thank you very much.
So, our next speaker this afternoon is Dr. Sandeep Pandit, who is going to be talking to us about the ionic basis of rotor formation and maintenance. Sandeep is a research assistant professor in the Department of Internal Medicine at the University of Michigan, and he has made some very important contributions on the mechanisms of rotor formation. And since today's session is on rotors, he is going to be telling us what a rotor is. So, Sandeep, you have the audience just very, very expectant to tell them what the rotor is. Thank you.
Thank you. So, good afternoon, everyone, and I would like to first thank the organizers and Pepe for inviting me to give a talk at Girona. This is my first visit to Spain, and I'm really loving your beautiful country. It's just gorgeous. So, thank you, Pepe. So today I'm going to be talking about the ionic basis of rotor formation and maintenance. So this is the outline of my talk. So in my background and motivation, I will introduce why do we study rotors and why do we care about the ionic basis of rotors. I'm going to talk very little about rotor initiation, but I'm going to spend a majority of my time on rotor maintenance and which are the more important ionic determinants of rotors. Right. So just to begin with, I think I'll skip for this audience. I need not explain why we study atrial fibrillation and why atrial fibrillation is important. But in terms of mechanisms of atrial fibrillation, I think we are still debating after a number of years what the different mechanisms of atrial fibrillation are. And in a slide review by Uli Scotton's group, I think he summarizes the different mechanisms that have been put forward that seem to maintain AF. So on top you could have possibly a single source that maintains AF. This could be either automatic focus or different kinds of rotors which are fixed or moving, or some other people are advocating the thought that it's not a single rotor, but it's multiple different sources that maintain atrial fibrillation. So it's not very clear right now. So what do we see in our lab when we study fibrillation, either atrial or ventricular fibrillation? And what we do is we really do experiments in isolated hearts, where we put an isolated heart in a Langendorf apparatus. We infuse voltage-sensitive dyes, and with the help of a laser and a camera, we can actually record the electrical activity on the surface of a heart, be it the atria or the ventricle. Now, when you are looking at ventricular fibrillation, atrial or ventricular fibrillation, the EKG, as you would expect, is completely chaotic. And what do we really see when you're looking at the heart? This is what we see. So what you're looking at is a movie in a rabbit heart, a rabbit ventricle to be specific in this case, which is in fibrillation, in ventricular fibrillation. And you can see, as you would expect from the EKG, completely chaotic activity. You see sometimes completely chaotic electrical activity. But this is not the entire story. So in a lot of times, this is what we also see. In this case, we were able to map a rabbit heart in ventricular fibrillation with two cameras, one on the anterior side and one on the posterior side. And this is what you see. What you're looking at is an optical movie without any processing. And what you're seeing very beautifully is a rotor that's going at the highest rate. And this is what is maintaining fibrillation in a number of cases. So this is the hypothesis that fibrillation, be it atrial or ventricular, is not always chaotic, but in a lot of cases, there is a stable single rotor that can drive this fibrillation. And it's not always possible to see this rotor. And this is not only in the ventricular fibrillation, but also in case of atrial fibrillation. So in this case, what you're seeing is optical movies recorded in sheep hearts. And you're looking at the left atrial appendage and the right atrial appendage. And if I can get your attention here, you can see that there is a very stable rotating activity in this heart that is maintaining atrial fibrillation. right? So again, this brings us to the concept that in certain cases there is a rotor that is driving atrial fibrillation. So rotors are now, people have started seeing rotors in atrial fibrillation, and this is very exciting. This is a paper or a figure from Sanjeev Narayan in San Diego, where he's recorded and shown that indeed these rotors can be present in human atrial appendages as well. So that brings us to some of the really basic concepts about rotors, which I'll introduce you to. So if you take a snapshot of a rotor on the left hand side, what are you really looking at? So this is what you're looking at really is something called as a wave activation or a rotor wave front. Now this is where the rotor is advancing and it is depolarizing the myocardial tissue. We call it as the wave front. At the end of the rotor, where the tissue is refractory, the rotor's end is defined by what is called as a wave tail. And the place where the wave front and the wave tail meet, this is the singularity point. So this is the wave front, this is the wave tail, this defines your rotor, and this is the singularity point. Now this also brings about a very important concept that is always put forward, what is called as the wavelength. But you see, if you think in terms of rotors, the wavelength is not completely or clearly defined, because the wavelength will vary depending upon, if you go closer to the singularity point, the wavelength gets shorter. If you go away from it, it gets longer. So wavelength is not a very clearly defined parameter if you think in terms of rotors. Now also, another thing that you can look at is if you define the singularity points in your experiments, in our experiments also, you can actually map how the singularity point travels. And that also tells us a lot of different things about rotors. And I'll tell you what kind of rotor properties we measure. The last thing to note about here also is you can look at action potentials at different points along this rotor. And what you'll notice is as you get closer to the singularity point, you can see that the action potential gets shorter and shorter. And this is because of more repolarizing current that is instilled by the core. So these are some of the concepts that are aligned with rotor theories. And if you look at a snapshot of a rotor on the right hand side, this is actually a snapshot of a rotor from a computer simulation where we can simulate these rotors. And what you're looking at is, this is the actual rotor where the cardiac tissue is depolarized. Actually, there's an action potential there. And in this deep blue area, this is where the cardiac tissue is completely refractory. There is no electrical activity there. And that allows us to look at what is known as an excitable gap. So in the panel below, you can see that what you're seeing in front of a rotor, this is what is generally termed as the excitable gap. And what you're looking at this is actually, what this tells us is what are the number of sodium channels that are available in this excitable gap. And that's how the rotor can actually invade this excitable gap. So you can actually, with simulations, you can quantify what is your excitable gap, how does your rotor look like, how does it meander in different places, and learn a lot about the different ionic mechanisms and their effects on rotors. Now, why do we care, or why do we study the ionic basis of rotors? And there are two main motivations. One is simply to understand which ionic currents are important in driving or initiating these rotors. And the second important thing, an obvious extrapolation from there is, if we can understand which are the main ionic currents, then we can start thinking about designing better antiarrhythmic drugs. And as we all know, there are two main problems associated with currently available drugs. One is efficacious, they simply are not good enough. Secondly, they cause proarrhythmia in the ventricle. So there are safety issues associated as well. Now, in terms of ionic currents in atrial cells, Ricardo in his previous talk really introduced us to different ion channels that are present in the atrium. And again, here is an atrial action potential, a cartoon of an atrial action potential that is driven by the sodium current, the calcium current, which are inward currents, and a number of different potassium currents that bring the action potential back to its resting stage. And the currents that are important, such as IK1 and the sodium-calcium exchanger, the last question that was put forward. Now, this gets much more complicated in chronic AF because, as both previous speakers have shown, the atrial action potential is shorter and there is a considerable remodeling in different ion channels. So we have to take this into account when trying to understand which are the important ion currents that drive these rotors. Now, let me spend a couple of slides talking about initiation of rotors. And in today's talk, I'm going to focus mainly on studies that have been conducted in our lab over the last 10 years, which have helped us understand some of the ionic mechanisms associated with rotors. Now, in terms of initiation of rotors, one very interesting mechanism by which rotors are initiated is called as vortex shedding. So this is a cartoon slide where it shows what the basic underlying concept is. So if you have an inexcitable object, this is depicted by red, and if you pace a tissue, and this is the electrical activation that's proceeding here. Now, as this activation wavefront proceeds here, it now starts to see this. Now this is your source, this is your sink, this is the wavefront that is going to proceed in this direction to activate the whole myocardial tissue. This is the critical point which will determine whether the rotor is initiated or not. And that is really dependent upon this radius or radius of curvature of the source. So if you look at panel B now, this is where we are activating a wavefront by pacing this site here, this star here. Now this wavefront proceeds here, and when it reaches this point here, it can do two things. If the radius is greater than a critical curvature, that is the radius here, what this rotor will do is it will align with this inexcitable object and then go back, and it will not detach. But if for some reason your radius of curvature is less than your critical curvature, this rotor won't be able to excite this whole tissue and it will detach. And essentially what we are looking at is a source to sink mismatch. So essentially, because of reduced excitability, this source is not able to excite this entire area in front of it, and that is how a rotor initiates here. That is, the excitability is reduced. And this was shown in a series of very beautiful experiments conducted by Candido Cabo almost 20 years ago. So in this case, what he did was they took a slab of sheep ventricular muscle in this regard. They created this inexcitable object. They paced the cardiac myocardium through here, and what you can see in red is the wavefront or activation wavefront here. And in the normal tissue, this wavefront proceeds along this and proceeds to excite the entire tissue. But when they reduced the excitability in this tissue by using TTX, which is a blocker of the sodium current, what they saw was that this wavefront now was detached. And you can see that this wavefront detached and it went without exciting this area, and these detached wavefronts now started beginning to form spiral waves. So vortex shedding is one very interesting mechanism by which these rotors are initiated. And as these experiments suggested, that reduced sodium current is what can create or initiate rotors. And this is what we actually see. So in our research study published just this year, last month, we find that the sodium current is indeed reduced in atrial fibrillation. And with the presence of inexcitable objects such as fibrosis, you can imagine a scenario where the rotors will be more easily initiated in atrial fibrillation. Now, let us talk about some of the different ionic currents that are responsible for maintenance of rotors. And I'm going to focus mainly on potassium currents, which we have studied over a number of years. And what we find are the different important potassium currents in maintaining fibrillation. Right. So first, I'm going to focus on the current that a lot of people have talked about, the inward rectifier current IK1. So how does this current look like? So if you take a patch clamp experiment and if you record this current, on the y-axis you're looking at the current density in picoamps per picofarad. On x-axis you're looking at the voltage. And what this current does is this current is very active in the negative region, and it completely shuts down when the voltage gets positive. So with this biophysical property, you can actually figure out how this current really acts in an action potential. So if you look in an action potential, this is an atrial action potential. It is very negative initially. It goes much more positive when you excite it, and it comes back. So based on this property, this is the sodium current that is driving this atrial action potential. So when this action potential goes more positive, this current shuts down, but it is active at more negative resting membrane potentials. Again, when the action potential starts coming back, this current becomes active, and it is important in the final repolarization phase. So IK1 is important in setting the resting membrane potential and in the final repolarization phase of the atrial action potential. So when I joined Dr. Halife's lab, they had done some beautiful experiments which demonstrated that IK1 was really important in maintaining ventricular fibrillation. So here I'm showing a slide which showed that when they induced ventricular fibrillation in guinea pig ventricles, these fibrillatory activity could be stopped by perfusion of barium chloride, which is a relatively selective blocker of IK1. So if you increased the concentration of barium chloride, VF was completely terminated. So this brought us to the hypothesis that, okay, IK1 is important in ventricular fibrillation. What about its role in atrial fibrillation? Is IK1 important? And we've conducted a number of experiments and computer modeling studies to answer this question over the years. And I'll give you some of the salient points that we have learned through these studies. Right. So the first study we started in this regard was with a computer modeling study, where we took a computer model of atrial action potential, which was able to simulate the atrial action potential in normal sinus rhythm, as well as its shortening in chronic atrial fibrillation. And this is very similar to what you see in patients, where the action potential is clearly shortened. Now, at that time when we started this study, it was also becoming very clear that IK1 was upregulated in persistent or chronic atrial fibrillation. And these were studies done by Dr. Dobrev and Dr. Van Wagener. So we decided to examine what does this do to the properties of rotors. So what we did was we created a computer model in a single cell where we generated three conditions. This was the control action potential. In red, you're seeing a condition called chronic AF1, where we simulated the downregulation in calcium current and different potassium currents. And in green, what you're seeing is another case where, in addition to these ionic changes, we instilled an upregulation of IK1. Now, what we did was, as you know, because the cardiac action potential propagates, it's not in a single cell, we put these single cells in a 2D tissue and simulated rotors. This is computer modeling that I'm presenting. And in the left-hand panel, you're seeing a case where we've incorporated a downregulation of different ionic channels, but no changes in IK1. And this is what you see. You can actually simulate a very stable rotor, just as I showed you in experiments. Now, what happens if you increase only IK1 and you increase its density twice, just as you see in experimental recordings in atrial fibrillation? This is what you see. Now, the same rotor starts going quite faster, and you can see it by eye, that just by increasing IK1, the rotor is going much faster. So with a computer simulation, you can actually go and try to understand why is this rotor going faster. So when we did that, before going forward, so two simple things, carrying on with the basic concepts of rotors. What you can do is, you can quantify how fast your rotor is going by frequency analysis. Is 5 hertz or 8 hertz? So you can quantify how fast it goes. The second thing that I mentioned earlier was, you can also quantify the meandering of the singularity point or the spiral tip. And by quantifying the meandering of that, you can understand how stable or unstable your rotor is. Because if the meandering point is very stable, it is likely that the rotor is going to be very stable. But if the singularity point meanders all over the place, it's likely that the rotor will extinguish itself by colliding with a boundary. So these are the two properties, I think, that are much better than wavelength to quantify, the frequency and the core tip meander. That helps us understand the rotor properties. Now, in this case, when IK1 was not increased, the rotor frequency was about 5.5 hertz. And this was the tip meander. You can see there's a large tip meander in the tissue. Now, when IK1 is increased, you can see that the frequency of rotation has gone substantially faster. It's quite up, 8.6 hertz. And also you can see that the meander, the rotor tip meander, has reduced. Or in other words, the rotor has become much more stable. It's stabilized a lot. So IK1 is doing two things, increasing the frequency and stabilizing the tip meander. Now, how does IK1 do this? So one of the very interesting things that we learned through our simulations is what IK1 does is not only it shortens the action potential, but more importantly, it makes the resting ionic potential more negative. And when you do that, you make more sodium channels available. And that is what is increasing this frequency. So actually, if you take snapshots of these rotors during the case where IK1 is not changed, that is, this is chronic AF1, and in the case where IK1 is increased, if you now look at the availability or the excitable gap or the availability of sodium channels, you can appreciate that about 60%, or in this case actually about 40% sodium channels are available. But in the case where IK1 is increased and the rotor is going much faster, if you now look at the excitable gap and the number of available sodium channels, you can see that this is about 60 to 70%. So this is one of the main mechanisms by which IK1 increases the rotor frequency. It makes more sodium channels available. And this was all nice. This was a computer simulation study. But does this really happen in real life? So we got an opportunity to test this in experiments. And this was a study done by a very good friend of mine, Dr. Simon RHYTHM, who's at Tufts now. So what we did was we were able to get our hands on a transgenic mouse where IK1 was overexpressed. IK1 was overexpressed 10 times. And you can see the consequence that this is a typical mouse ventricular action potential in a wild-type mouse. But in the transgenic mice where IK1 was overexpressed, the action potential was much shorter. And we could induce experimentally very fast rotors in these transgenic mice. So on the left-hand panels, I'm showing you, these are experimental data, optical movies. You can see that we could see rotors about 30 hertz in wild-type mice, and these were unsustained rotors. But in transgenic mice, where IK1 was upregulated, we could see very stable and very fast rotors of about 55 to 60 hertz of frequency. This is, of course, the mouse, the action potential is much shorter, so the rotors go very, very fast. And when initially Sammy saw these results, both Pepe and I, we did not believe these results. But we actually said, why don't we take your results, put together a computer model and see, can we actually simulate this? And yes, we could. When we actually put together a computer model and just increased IK1, just as we see experimentally, you could see that you could simulate a rotor, slow rotor about 20 hertz and a very fast rotor about 47 hertz just by increasing IK1. So this was a very good complementary study to give much more proof that yes, IK1 is really important in driving these rotors. So these studies told us that yes, IK1 is a key determinant of rotor dynamics in terms of the frequency and stability. Now the logical question was, okay, if this is the case, can we start thinking about designing antiarrhythmic therapy that is targeted towards IK1? And yes, this is what exactly has been done in the last couple of years. So in a very beautiful study conducted by David Felgueiras, who's sitting with us today, so he used a drug called chloroquine, which is an anti-malarial drug, but which is also a more potent blocker of IK1. Chloroquine blocks a number of different ion channels, but it is a selective blocker of IK1 as well. And indeed, what I'm showing you here is this is a case of paroxysmal AF, in this case, stretch-induced AF. And you can see on the left-hand side that you can have AF of about 9.6 hertz in this case. And when you perfuse chloroquine, it reduces the frequency, and every single case, chloroquine terminated atrial fibrillation. This is again acute atrial fibrillation, so we have to see if chloroquine is effective in persistent atrial fibrillation as well. Now let us look at some different ion channels other than IK1. I've already shown to you that IK1 we think is really important in driving atrial fibrillation. What are the roles of other different potassium channels in fibrillation in particular? So we've studied a number of ion channels, and iCAL, or the fast delayed rectifier current, is one of the principal repolarizing currents in both the human atrium and the ventricle. So if you actually block this channel in the human atrium, what you will see is that the APD is prolonged, and this is in normal sinus rhythm, and including in atrial fibrillation also, iCAL block seems to prolong the APD quite a lot. Now, if you induce AFib and if you use dofetilide or E40301, or in this case sotalol, and measure the AF cycle length, indeed the AF cycle length is increased when you block IKR, and you can expect that the frequency of rotors will go down. This is a study conducted by Stan Nettel. But in our lab, we took a different approach. We decided to study the role of IKR by studying rotors in neonatal rat ventricular myocytes. So this is a very beautiful model in which you can study rotors in a very consistent form without any structural heterogeneities. So what we did was we essentially overexpressed IKR, or the hERG, the molecular basis of IKR, and you can see when we overexpress this with the help of an adenovirus, the current density of IKR is increased compared to controls. And now, if you put this in a neonatal rat ventricular monolayer, what you will see is, in the case of GFP, you have about 10 hertz. When you overexpress IKR, the rotation frequencies increase substantially, about 20 hertz. And if you now suppress IKR, this frequency goes down, so about from 10 hertz to about 7 or 8 hertz. So indeed, we can see that IKR also is important, but it doesn't seem to accelerate the rotors as much as IK1. And most interestingly, we could look at what are the mechanisms by which iCAR is accelerating these rotors. And turns out, again, it follows the same concept that it shortens the action potential. And very interestingly, we found that it also causes a transient hyperpolarization, just like IK1, but only in a transient nature. So these are the two mechanisms that help iCAR drive the rotor faster in terms of fibrillation and rotors. Now, what about IKS? I think Ricardo presented us some very interesting data that IKS is probably upregulated in chronic atrial fibrillation. Now, IKS, it turns out, at least in a normal action potential, this is a ventricular action potential, it does not contribute much to the action potential. So IKS seems to be more important during adrenergic stimulus. So we use the same approach of overexpressing IKS through adenovirus in neonatal ventricular monolayers. And what did we see? So this is again showing you data where we show that this is GFP, and if you over increase IKS current, you can get a larger current density in neonatal right ventricular monolayers. Now, if we look at rotor frequencies, it's very interesting. There is a trend for increase in the rotor frequencies, but the rotor frequencies are not significantly different if you overexpress IKS. In contrast to IKR, remember IKR, if you overexpress, the rotor frequency was faster. But it turns out that if you overexpress IKS, the rotors don't seem to go faster in this case. What you do see instead is these kind of wave breaks. So if you follow me through the cursor, you will see that at 0 seconds, this is a rotor where there are no wave breaks, and this is giving activation wavefronts. Now, if you follow it around time, around 4 minutes, around 6 minutes, this is the case where there is control and there is no overexpression of IKS, and you can see that there is a stable rotor that gives us activation wavefronts. But in the case where IKS is overexpressed, you can see that you can start seeing these different kinds of wave breaks. And by let's say 5 minutes or so, you can see a number of different wave breaks. And this is what is quantified in the panel below, that you start seeing, as time proceeds, you start seeing a number of different wave breaks. They increase over time when IKS is overexpressed. The frequency doesn't seem to change much, but the number of wave breaks start changing. So we looked at what is the underlying mechanism for this, and in this case we conducted a study in a simple HEK cell where if you express KIR 2.1, which is the molecular correlate of IK1, and KVLQ-T1, which is the molecular correlate of IKS, and you can actually do a current clamp and simulate action potentials, and this is what you find. If you do a S1, S2 protocol with a large S1 duration pulse followed by a short S2 pulse, and this S2 pulse is advanced closer and closer to S1, this is what you will see. If you only express KIR 2.1, which stands for IK1, you'll actually get an action potential like waveform. So this is due to the negative rectification characteristic of IK1. You will generate an action potential like waveform. And if you bring the S2 stimulus closer, you can see that you can generate a similar action potential like waveform, which is of shorter duration because the stimulus is shorter. Now, what happens when you express IKS along with IK1? What you'll start seeing is this sort of, you cannot, as you bring the S2 stimulus closer and closer to your S1, you cannot evoke this action potential like waveform. This is because IKS is overexpressed, and what is happening is this overexpression of IKS causes the current to die out slowly, and what you get is what is known as post-repolarization refractoriness. And this was shown by Pepe about 30 years ago in guinea pig ventricular myocytes. And this is what is exactly happening in this case. What is happening is because of overexpression of IKS, you're creating what is known as post-repolarization refractoriness, which is giving rise to what we think are different wave breaks. Right. So what about the other potassium currents? So the other main important potassium currents are ITO and IKUR. And in our case, we have only studied them through simulation so far. So if you block, in this case, by AV0118, which is a selective blocker of IKUR, you'll see that it prolongs the atrial action potential in normal sinus rhythm. Yes, almost there. And it prolongs the action potentials along in the plateau phase as well as APD90 in chronic AF as well. And what we see is, at least in computer simulations, if you block IKUR or if you block ITO, they seem to actually terminate rotors. They don't change the frequency much. But interestingly, the way they terminate these rotors is they increase the tip meander. So apparently, at least in computer simulations, if you block iCAL and ITO, the tip meander of the rotor goes haywire, and then the rotor terminates by colliding with the boundary. But we have to verify this in experiments yet. These are just computer simulations. So to conclude, what we have seen is initiation of rotors. There are many mechanisms by which you can initiate rotors, but vortex shedding is one of the important mechanisms, and reduced sodium current is an important ionic basis by which a rotor could be initiated. A reduced sodium current does occur in atrial fibrillation. IK1 seems to be a very key ionic determinant of rotors in terms of their frequency, how fast they go, as well as their stability. IKR seems to affect rotors in terms of their frequencies, but interestingly, IKS, in our hands, seems to mainly affect these wave breaks or fibrillatory conduction. And the role of IKUR and ITO is less clear, and we need many more experiments to understand their role in maintaining rotors. So with that, I would like to thank all my colleagues and mentors. Thank you very much.
Very good, Sandeep. Thank you very much. Questions or comments for Sandeep? Should I ask you a question, Sandeep?
Sure.
Any questions? Come on, the clinicians, come on, ask questions. Make comments. How much does this relate to what you're doing? Rihanna, please.
Yes, thank you. I would like to know, because I didn't understand very well, the rotors are more surface dependent or more volume dependent. In other words, which is the minimal tissue in which in a human atrium can you observe a stable rotor?
So I think a number of years ago, and I showed you some movies, for example, from the mouse heart as well. So you could clearly see that you can sustain a rotor even in a mouse atrium or a mouse ventricle. So I think the issue is not exactly the amount of tissue, area or volume. It's actually the excitability. If your APD is short enough, you can even induce sustained rotors in a mouse. And in terms of talking about surface, I think you can see rotors on the surface as well as you can see intramural rotors as well. So I think from our lab, Jerome Khalifa has shown intramural rotors in the atrium as well. Does that answer your question?
So let me expand on that, Julian. The ventricle is very thick, and sometimes if you're recording from the epicardium, you will see a rotor. Then if you go into the endocardium, you may see the rotor because it's three-dimensional. It's a scroll. It's not just a surface spiral, it's a scroll wave. So they do span many times the entire wall. So these are three-dimensional objects, what you're looking at, but we represent them in two dimensions because it simplifies space. But it is an oversimplification of a very, very complex problem because those rotors that can have a center of rotation that is like an eye, can also have a U shape, can also have an L shape, and it can have a very complex shape that spans the ventricle. So it is very complex, but we are making things simpler so that in our mind we can understand them better. But that's a very important question.
Yes, but we try to understand the mechanisms because the wavelength was very nice theory. And in practice, could be successful if you perform a maze approach, even in chronic or permanent AF. The patient could maintain sinus rhythm. So, talking about rotors, we would like to put boundaries, and at least we would. I would like to have an idea about if you can tell me in 1 centimeter square you can maintain 100 rotors. So I'm not going to burn anymore because that means that 1 centimeter square of atrial tissue can maintain AF. But if you treating the patient with antiarrhythmic drugs, for example, as happened with the theory of the wavelength, you could expand the cycle and maintaining the same amount of tissue, that would be, theoretically, the explanation.
Yeah, I think you don't need to think that in terms of wavelength. You need to think in terms of frequency. And that is something that clinicians have really not thought about. The cycle length of atrial fibrillation is a very important parameter that needs to be addressed if you want to think about rotors. The fastest rotor is the one that predominates. And you're going to find the fastest rotor in a small area. And if you can find that small area, that's what Felipe and the radar investigators were trying to do. And they were successful to a certain extent. They were just as in atrial fibrillation, they were just as successful as with the isolation of pulmonary veins. So it is frequency, the parameter. Frequency, as Sandy was saying, meandering. The problem of wavelength is that you don't know where to measure that wavelength. And that has been the problem. If it is an anatomical obstacle, wavelength has a meaning. But if it is a rotor and is meandering around, wavelength has no meaning. So you have to look at frequency. Can we, you guys want to have coffee or you want to just one very fast, please.
Hello, this is Jose Guerra from Barcelona. I know that when talking to you guys, we are all the time talking about electrical remodeling, but what is the role, if there is any, for structural remodeling? Because is fibrosis, anisotropy, it has any role? It's just an anchor point, or what is it going?
Your question is the segue for the next session, so stick around, please. Let's have a coffee break. We'll come back in 20 minutes, please, at 10 after 5. Very nice, Andy.